Hi everyone, I am Saniya and in this video, I just want to talk to you about GProfiler, which are web tools for functional genomics, interpretation, and analysis as well. So I prepared a few videos on GProfiler tools, which are very powerful. But here, I just kind of want to talk to you a little bit about what each of the tools are doing and why they're important. And again, I have a more in-depth videos and examples kind of walking through using each of these tools. So I just kind of want to get started and show that to you guys. And please let me know if you have any questions. And again, please note that I do have extensive videos on each of these examples. But here I just kind of wanted to give an overall snapshot of GProfiler and what are the tools and why are they important as well. So here we can see that this is the GProfiler interface. So the URL for this is the following. I'm just going to paste the URL here. This is the URL for a GProfiler. So they have a URL version and they also have an R version that is available. So you can run this in R, and I'll be preparing some videos about running this in R as well. But they have some various options. And again, I have videos for each of these. But what you can see is that we have GOSt, which is functional profiling as well. So it does some enrichment analysis. So we can even look at a mixed query example as well. We could just run this, for instance, and we can see some of the detailed results that we can get. We can get all these various results with different software packages, database packages like Wiki Pathways, React, KEGG pathways, gene ontology, cellular components, gene ontology, biological processes, gene ontology, molecular functions. And again, I have more extensive videos on this. We can also just have a random example, run the query, have various enrichments that we receive from here, functional profiling, which in other words is just trying to help us do overrepresentation analysis, mapping genes to known functional information sources, and detects statistically significantly enriched terms from various data sources like gene ontology, biological pathways, regulatory motifs in DNA, protein databases, and human phenotype ontology. Then next, what we have is we have gene ID conversion, which is helping us convert between our gene protein microarray probe and numerous other types of namespaces. So you can go from ensemble to entree gene IDs. It's just a really good reference that way to go from one type of identifier to another. So you can go from a target to, you can go from certain numeric IDs that you have, and then you can map them to ensemble IDs or entree gene IDs. So you can map them to various types. So if you want to map between different IDs for a given gene. And what's key is that, for instance, if we check out like the APO gene, this is a really key apolyprotein gene for Alzheimer's disease. For instance, you can see this is HGNC IDs, NCBI entree IDs, ensemble IDs, OMIM. So there are various IDs, and then you can try to map between these. And of course, there are so many more types of IDs out there for genes, so many different databases and nomenclatures that you can use. So that's also key. Then I wanted to show that that's another option that you have here. There's also GORth, which is orthology search. So you can translate gene identifiers between various organisms. So I have a video where I talk about having 59 different genes, and they are represented by their entree IDs. And again, an entree ID is just one of the identifiers. So I was trying to represent them by these entree gene IDs, like for instance, APO's ID is 348 entree ID. And I'm trying to map them between various organisms like mouse, mus musculus, the cows, and cats, because I have the two little cats, cute cats, as you guys know. And I'm trying to map between them and then determine which are orthologous gene mapping. So genes from one organism to another organism, like what are the similarities between them, what genes tend to map and have similarity between them. And then lastly, this is SNPense, the SNP ID to gene name. So if you have a bunch of reference SNP IDs given by these RSIDs, and then you can run this and figure out which regions they map to, in the DNA, which is really key because SNPs do have an impact. They're single nucleotide polymorphisms, and this mutation has an impact somewhere in the DNA. And you might want to figure out, well, which genes do they link to, and what are the predicted impacts that we now know about those. So that's sort of like how you can use these 4 key tools. And for each of these tools, I had prepared a video for each of these to walk through and kind of explain the analysis and the output for that. And the way you can also see is that it's just basically a tool set for functional enrichment analysis and conversion of gene lists. Because biological data analysis, it often deals with lists of genes from various studies, and GProfiler is widely used for finding biological categories enriched in gene lists. This part is done by the functional profiling part. It's used for this, used to convert between gene identifiers and mappings to their orthologs. This part is given by the conversion from gene IDs from one group to another for humans or for other organisms. So this allows you to basically convert between one ID type to another ID type for genes for one organism. That's what this is allowing you to do, gene ID conversion. Whereas this orthology search is basically gene ID conversion from one organism, so not one type to another type, but from one organism to another organism. That's what this is allowing you to do. You have these genes. And then lastly, what is really cool is that this is this SNPense, which is basically like, how do you make sense of your SNPs? So then you have the mission is to provide a reliable service based on up-to-date high-quality data in a convenient manner. So it uses data from ensemble database, fungi, plants, metazoa, specific version of ensemble genomes, and all of this. And they use data from KEGG, Reactome, Wiki Pathways, Transfac, MI, Tarbase, Human Protein Atlas, CORM, and HP. These are various databases that are out there. So over 500 organisms and hundreds of identifier types. And they even have this in R. So kind of just going through again. So this GOSt sounds like a ghost. It performs functional. So this first part, this is performing functional enrichment analysis, also known as overrepresentation analysis or gene set enrichment analysis on input gene list. So you just have to give in some ensemble IDs. And again, these are one type of ID because for APO, for instance, we have this ensemble ID that is representing it. And as you can see here, we have these ensemble IDs in this example here. And again, I talked more about that in that video. It gives a plotly graph with the, with Manhattan plot similar to the one shown by the web tool. So an example is that when you run this query, the plot is interactive, so you can hover over it. This is generated using Plotly, which is really cool. It's a really cool data visualization kind of a tool set for making interactive hover over those type of things. Then GProfiler Convert, which is this one, gconvert. Its job is to help you convert between various gene, protein, microarray probe, and numerous other types of namespaces. So if you have one type of ID, how do you essentially translate to another type for a list of genes? Like if I just have this ensemble ID, how do I figure out the entre ID or vice versa? That's what it's going to allow me to do if I use the convert. Like if I just have a list of these IDs, how do I find out these IDs, for instance, for those same genes? And then GProfiler Orth, gene identifiers between organisms. So this is for within one organism, different types of IDs. This is across organisms. So that's what this tool is doing here. And then GProfiler SNPense is mapping SNP RS code. These are reference SNP IDs to gene names, chromosomal coordinates, and variant effects. So that's just helping us, again, make sense of this list of reference SNPs to their effects here. So if you look at this, for instance, you have a set of genes. We have this plotly graph where it's showing like, okay, this is the gene ontology, kinase activity. This is the negative log 10 adjusted p-value. These are data sources that support this finding that you have here. I think it's showing this plotly graph here. You have the color-coded evidence codes for gene ontology that sort of are generated right here. Then what you can also do is, like, let's say you have a canine, a dog, you have a target species, you can do gene orthology search as well. If you have a list of, let's say you have a list of this is the mixed effect query that they give you an example. You can run a mixed query example. And this example here is sort of what they're showing over here. Like, let's say you have this random query and we run this mixed query example here that they're showing. So all these rows are the same as over here. Then you're able to basically figure out, you have a plotly of the results here for this set of 5 inputs. You have this plotly of results here for the molecular function, 33 molecular functions, 111 biological processes, 21 cellular components, 1 KEG, 8 React, 3 wiki pathways, 1 TF, mRNA, HPA, CORM, HP. So these are different data sources here. And then you can convert these, let's say, to different target namespaces using gene convert. You can convert these as well. So we can take this and then do some conversion. So that's what's really cool, that we can just take this alias and convert them to onto a gene ID using gene convert. That's sort of what this example is showing you, how you can convert them to ensemble IDs. So from this, you can convert them to ensemble IDs from this list of points here. You can convert them to this. You can also figure out their functions, which is what we were doing here with the functional profiling. We can have a set of detailed results here for them. You know, angiogenesis, regulation of synapse activity. We have all these results here. Glutaminergic, synapse, all these. That's what we can see here. We can also do orthology with canines, for instance. So if we can go through orthology search and map them from humans to, let's say we want to look at canine. Canis lupus familiaris, dogs. And then we can map these aliases and the ensemble IDs to the ortholog. So we can go here from humans to like dogs, for instance. And get some description as well. And that's pretty cool. This is dogs. And we can map them as well. So we can get different identifiers. We can map from human to dogs for this as well, which is pretty cool. To go from one organism to another using the Orth part here. So we can do the ID conversion from onto IDs to ensemble. We can do that conversion here, figure those things out. So we've kind of talked about so far how we can get the functional impact from GOSt, how we can do the target species, how we can do the target namespace, and then if we have some random set of SNPs, how we can predict the variant effects as well. There's a lot as well that we can do in this, and accompanying paper with more information on this that I can talk through some more. But I also wanted to show you that there's also this interface in R that we can use that was just updated a few months ago. It's a GProfiler 2 package as well. And it's sort of like using kind of this GProfiler. It has the GOSt functional enrichment analysis, the GConvert, GOrth, and SNPense as well. So a lot of tools that you can use here, and you can run these in R, GConvert. You can get a lot, GOrth. That's using the GOrth tool that, given a target organism, retrieves the genes of the target organism that are similar in sequence. So it looks at sequence analysis like evolution and tries to see essentially like what maps between us and another organism. So it's trying to do that predictive analysis, like what are similarities between them. And then what the next thing that we could try to do is we could, so like for instance, it shows you that, you know, you have a source organism like humans, you have a target organisms like mice, you have a query. A query is a character vector of gene IDs to be translated, and what are the ones that are overlapping. So it's this idea of GOrth is the same idea as like kind of going here having a list of genes and just trying to map them. All these to the ortholog name and ensemble ID and description. So you could see that over here as well. They have all these. They even have a GOSt plot, which is the Plotly Manhattan plot of the results. It's very similar to the one shown in the web tool. But here you can also check this out as well. There are a lot of functions here, and I'll make some more videos going through each of these as well, hopefully, if you find that helpful. And they also have like this description as well of kind of a vignette. I'm a huge fan of these, especially because they usually have the results. Like this same thing here that you'll see is the same as like this exact idea here. 1 second. I think except for the 3837, but they tend to have the same idea here, geo and then this. So that's like an overlap here. And then you can see in R, if you run the results, what these look like here. Visualization of the functional enrichment results. These plots, how to interpret them. Target numbers. So this is a lot of resources out there. But yeah, this is just how to make sense of all the data that you have. So I hope that this will be helpful for you guys. So if you found this helpful, like please, let me know. Please like, please subscribe, please share. If you have any questions, please let me know as well. Write any comments, questions, concerns, and any advice as well. So I'm Saniya, and I hope that this was helpful for you guys. Thank you and have a great day. Please do see the description bio for the links for those other 4 different tools available in GProfiler. So yeah, again, those are very extensive walkthroughs of each of the tools. And here's just going to be a very quick overview about GProfiler as well. And I'll be having some more videos kind of talking through the R programming aspect of GProfiler. But we're going to just dive in right now.